Hello, I'm Oscar Bastidas. I'm CEO of Binartis Genomics, and today I'm going to answer some questions about genomics. These questions are coming from the eTwinning school, a community of European schools. Although genetics can be very complex, I will try to keep it simple so that everybody will be able to understand. So what does your work consist of, or what does a genomic manager do? Well, a genomic manager makes sure that the genetic analysis is properly done, and these are all the steps of a genetic analysis. First of all, we have a genetic consulting, then we extract a DNA sample. A sequencing machine extracts the DNA data from the sample, and then the data is treated using computers, and then a genetic analyst performs an interpretation of this data and generate a report for the doctor to be read. So in the first step, the customer or the patient understand what is possible and what is not possible with a genetic test. Genetics is complex and not everything is possible. It's not like any other type of analysis. Here, after that, the sample, we make sure that the sample has a good quality so that the DNA can be extracted. If the saliva or the blood are in bad condition, it cannot be analyzed. In a second step, the DNA information is extracted with a machine that is called a sequencer, and after that a second type of analysis is performed with computers. This is called bioinformatics. After the bioinformatic analysis, a genetic analyst checks all the variants or mutations that you have, that the person has on their DNA. This means the changes on the DNA that can be a cause of a disease, and after they prepare a report for the doctor. Finally, this report explains the results to the doctor or to the customer so that the genetic data can be understood by the person that is reading the report. So why did you want to be a genetic analyst or a genetic counselor? What made you choose this job? Well, my personal motivation is to help people. This is one of the reasons I choose this job. And it was also very interesting for me that I was able to save lives. I think it's very rewarding to be able to save lives, and also it's a real exciting challenge. Genetics is advancing a lot in these days. In the last years, in the future, is going to advance a lot more, and this is a real challenge that is very exciting area to be working on. What is digital genome engineering? Okay, so if we forget about the word digital for a while, what is genome engineering? The genome is the totality of a DNA of a person. The genes have the information that make us to be as we are. So if we have white skin, blue eyes, this information is stored in our genes. That means that if we are able to change our genes, we could change these traits. We can change the color of our skin or the color of our eyes. That means if we could cut a small portion of our DNA and replace it with different information, this can change the human being that we are. There is a technology that has been developed recently that is called CRISPR, and is similar to a pair of scissors, and that allow us to cut portions of the DNA and replace it with different characteristics so that we can change living organisms. So does it mean that we can change the DNA of a person? If a person is fat, we can change it to become thin and strong. Or if a person is sick, we can change the DNA of a person to become healthy. Well, yes and no. All the cells of our body share the same DNA, and it's very difficult to change only a few cells to cure a disease. So the easiest way to do this is to change the DNA before the person is born, so that we can change the DNA of all the cells at the same time. Right now it's quite difficult to do that, especially in humans, but it's not impossible. But the thing is, to change the DNA of a person to become stronger, that would be illegal. But if we change the DNA of a person that is sick to become healthy, then this is legal and it can be authorized by the authorities. So the last word is digital. Digital genome engineering is, in order to perform all this genome engineering, if we do it with the help of computer, that means that it's digital. So that will mean to change the DNA with the help of computers. That is digital genome engineering. So another very interesting question is, how can genetic diseases be prevented? Very good question. So most of the time genetic diseases are due to bad habits like smoking, drinking, also because of exposure to sun and eating bad. So the best way to prevent these diseases are not smoking, not drinking. Well, almost everybody knows that. Also, especially if you have a white skin, if you expose yourself too much to the light of sun, it can be damaging. It can provoke skin cancer. So another question is, what is the hardest part of your job? So one of the things that I find difficult is sometimes in genetics things are not clear. You can have a person that is sick, and we analyze their DNA, we have a report, but the report doesn't have a clear result. There is a lot of uncertainty and we are not sure what is the genetic mutation that causes the disease. Sometimes this is because not always a genetic disease can be in their DNA. 30%, this is roughly 30% could be of genetic origin, and 70% can be because of environmental factors, like things that the person has done during his life and they are causing the disease. So another question is, what tools do you use in your work? Well, these are the kind of tools that we use. This is a pipette that is usually employed in labs, in laboratories for genetic analysis. This is a centrifuge machine that is used also to treat the sample and to extract the DNA. This is probably the most important machine. This is the sequencer. This is very expensive, and this extracts the DNA from the biological sample. And then computers, we use a lot of computers for data interpretation and also to check in the databases what are the relevant information from the DNA. The databases look like this. You can see the DNA and how each mutation and how each change in the DNA affects the human that has this mutation on his DNA. So what studies are done in genomics? Well, in genomics is a fairly recent science, and the human body has more than 20,000 genes, and we still don't know all the functionality of every gene. So one of the most important studies that we perform, the scientists do, is to find out what is the function of each gene. And another type of study is to figure out if a medicine will work on a person depending on the genetics of this person. There are specific mutations that if the mutation is present, the medicine is not going to work. Whilst if the mutation is not present, the medicine will cure the person and the medicine will work. So this is called pharmacogenomics, and it's also a very important study that is performed in genomics today. So is the degree very difficult? How long does it take to study? Well, I cannot say if it's difficult or not. I didn't study genetics. My training is telecom engineering. But what you need to be able to work as a geneticist is a life science degree, 3 years after high school, such as biotechnology, biology, biochemistry, or genetics. Clinical genetics is not available yet in our country because some legal issues, but that will be the bare minimum that you need. After that, it could be also convenient to have a master degree in biomedicine or a master in genetics. That will be 1 more year, but it's not mandatory. And in case you want to do research, that will be 3 more years if you do it full time, or 5 years if you do it part time, and that will be more convenient for research to be able to investigate and do research on the latest things in genetics. Okay, another question is, how do you analyze the tests? I explained a little bit before, but I'm going to focus on what we do exactly. When we take the sample and we detect what are the mutations that we found on the DNA, what we do is we check these mutations against public databases with clinical information, and in many cases these mutations are reported in these databases, and if we find it in one database, we can already say that the chances of this variant to be pathogenic is high, and if we don't find it in many other characteristics are met, we report it as a benign or a low probability of pathogenicity. And this is basically what we do, is to check on public databases most of the time. Sometimes are private, but most of the time are public. What are the chances of this variant to be pathogenic? So, do you recommend this job? Yeah, myself, yes, I do recommend this job because of many reasons. It's very nice to help people. From time to time, we have the chance to save lives, or at least to provide information that helps doctors and people to save life. To me, it's a really exciting challenge. It's difficult, but once you have interesting results and you are able to help people, it's very rewarding. It's also a growing market, it's new, and medicine and health is always important in the public and the private sector. It's something that interests all of us. All of us want to live longer and to live better and be healthy. So it's a very big market because of that, and the satisfaction to help people to do that, for me, is one of the best things of my job. Okay, another question is, what is the education period and the content of the genetic and biomedical engineering department? Biomedical engineering interests me more. Is it possible to work in coordination with genetic engineering? Yes and no. I mean, everything is possible, but it's going to be a bit difficult because biomedical engineering has to do more with hardware, like machines or devices that are going to help patients in a hospital or in a clinical environment. Whereas genetic engineering is more about modifying the DNA most of the time of plants, because what we discussed in another question. Genetic engineering with humans is very limited, and even for research it's difficult to do genetic engineering in humans. So to me it's quite different, two different disciplines: biomedical engineering with the hardware component, and the genetic engineering that is more related with DNA pipetting, is more a clinical lab work, a classical lab, where you're going to be with tubes and pipettes and everything that has to do with liquid and also with computing, but it's not machines or devices. So how can genetic diseases be prevented? This is a very good question. Well, it depends on the disease, but as a generic rule, there are a few things that can affect most of the disease that are understood to be bad if you do it. And so the better way to prevent genetic diseases without knowing which disease is not smoking, not drinking, or drinking moderately, to prevent lung cancer, liver cancer, and many other diseases. Avoid too much sun exposure, and practice sports and have a healthy diet. Avoid processed meat or excessive red meat and everything that has everything that is unhealthy. So for a specific disease like breast cancer, this is the kind of things that we do. We do a genetic analysis, and in case we detect a predisposition to suffer, for instance, breast cancer, here we can do things like preventive medicine to increase the level of examinations, like instead of doing it after 50 years old, a woman can decide to perform regular exams after 30 years of age and every 6 months instead of every year. That is the regular, I think is the regular period where women can do. Also stop smoking and any other good habit we can do that we know that already is one of the external factors that helps cancer to develop. So how do you investigate the DNA if it's smaller than a cell? This is a very good question also. It's very small. It's very difficult to see it even with a powerful microscope. So the technology that is used, first, where we extract the DNA from cells with a procedure that is quite easy. And once we have the DNA, like the double helix, the helix is called denaturated, and it means that it's separated from the other part of the helix. And in a very simple way, we attach probes that are like little pieces of small lights, microscopic lights, that are going to bright with a different color depending on the nucleotide, the letter that they attach. In this picture, if we attach to a G, it's going to bright in a yellow color. And afterwards, it attached to a T, it's going to bright with a green color. A is a red color, and like this, with 4 different colors, we can sequence the DNA of any cell or human body. To sequence is to extract the letters of the DNA, to make it simple. And this is the technique. There are other techniques, but this is one of the most popular ones, is to use microscopic lights to detect what are the nucleotide, the letter of the DNA. So another question is, can diseases be cured through genetics? Well, the answer is yes, but with gene therapy, which means that we are going to work on the product of a gene or to substitute the function of a gene somehow, introducing RNA or something similar. But this is very rare. It is only used in a very few rare diseases. It's not very common. The most common way to help people with genetics is through better diagnosis, because genetics is a very precise way to detect where a problem is. Whenever we have a mutation on a gene, this gene is probably a very nice piece of information for the doctor, for the medical doctor, to decide what treatment to give to the patient. And thanks to this information, it can help much better to cure the disease. But Genetics doesn't cure by itself. It's a tool that helps doctor to cure the disease and provide very useful information. So thank you. I hope I answer all your questions successfully. If you have any other questions, you can send us an email to info@binartis.com, or you can also, if you are watching this on YouTube, you can leave a question in the comments and we'll try to answer as quick as possible. Thank you.